Good day. My name is Dr. Abdulazeez Azeemat from the Department of Biological Sciences, Al-Hikmah University, Ilorin, Nigeria. I'm here to make a presentation on a research titled Pharmacological Mechanism of Antiulcer Activity of Chromatographic Fraction obtained from Acacia nilotica Seedpod in experimental rats. Introduction. Peptic ulcer is a very common disease that is characterized by ulceration on the epithelial lining of the stomach and duodenum. If the ulceration is on the epithelial cell of the duodenum, it is generally known as duodenal ulcer. But if it is on the stomach, it is known as the gastric ulcer. But altogether, they are known as peptic ulcer. This disease affects about 10% of the world population. And there are lots of orthodox drugs that are used for this treatment of peptic ulcer. Although most of these drugs have brought a lot of remarkable changes, their efficacy is still debatable because of so many side effects and the availability of these drugs. Medicinal plants and their derivatives have been an invaluable source of therapeutic agents to treat various disorders, including this peptic ulcer disease. Acacia nilotica is a common tree that is found in Africa, including Nigeria, and different parts of this plant has been used for different therapeutic purposes. Its seed pod is used here in Nigeria to treat peptic ulcer by soaking it inside water, allow it to boil, and the concoction is taken with milk for the treatment of peptic ulcer. The objective of this study is to fractionate the aqueous extract of this Acacia nilotica pod to evaluate the anti-ulcer effect of the chromatography fraction obtained from these Acacia nilotica pod, and also to elucidate the possible mechanism of gastroprotective action of the most effective fraction of Acacia nilotica pod. The materials and method. This study was carried out after ethical approval from the University of Ilorin, Nigeria Ethical Clearance Committee with the attached approval number. The aqueous extract of Acacia nilotica was prepared by macerating a known gram of Acacia nilotica seed pod in distilled water and for 3 days, that is for 72 hours. This was filtered using a muslin cloth that was later refiltered using a filter paper, and the extract that was gotten was lyophilized. The product that was gotten were then subjected to solvent-solvent partition using different solvents of increasing polarity. We use N-hexane, ethyl acetate, and N-butanol. At the end of the day, we had extract that contained ethyl acetate extract because the N-hexane extract was very minute and we could not use it for anything. So we had only the ethyl acetate extract and the N-butanol extract and the residual aqueous extract. The N-butanol partition fraction that was gotten from this aqueous extract was later on subjected to column chromatography as described by Dito, and this was the scheme that was used for this extraction. The aqueous crude seeded extract that was used from aqueous elution were reconstituted using distilled water, and these were then extracted with N-hexane. After that, the N-hexane soluble fraction was collected, and the remaining aqueous solution was re-extracted using another solvent, which is the ethyl acetate. The ethyl acetate soluble fraction was removed, and the remaining aqueous fraction was re-extracted using N-butanol. So these are solvents of increasing polarity. This is the solvent-solvent partition. So the N-butanol fraction was then subjected to column chromatography. This column chromatography, we use mobile phase of formyl ethyl acetate, and at the end of the day, we were able to pull up 3 different fractions labeled as fraction A, fraction B, and fraction C. So the 3 fractions that were gotten, which is the fraction A, fraction B, and fraction C, were subjected to biological assessment for anti-ulcer effect. 36 animals were randomly grouped into 6 groups, and they were given to protocol in which group 1, we had it as controlled. That is the group that was not ulcerated, and at the end of the day was only given distilled water as treatment, while we have the group 2 in which they were given distilled water, and at the end of the day, they were ulcerated. Then we have the reference drug that was ulcerated, and the treatment group using 5 milligram per kilogram body weight of each of the fraction, which is the fraction A, fraction B, and fraction C. This treatment was done for 7 days. And on the last day of the experiment, just about 1 hour after this treatment were given, all the groups that were supposed to be ulcerated, which is the group 2 to group 6, were given 1 ml of a 95% ethanol orally in order to induce gastric ulcer. So here, ethanol was used as the ulcer-inducing agent. Immediately after an hour after this, the stomach were removed, they were cleaned, and the gastric juice were removed, and the ulcer index were calculated by measuring the amount of blood streak that were found on the gastric mucosa, and they were all added up together to give us in millimeter, to give us our index in millimeter. Then at the end of the day, the experiment showed that fraction B had the most gastroprotective effects, and thus the next experiment was the evaluation of the probable mechanism of action for the anti-ulcerogenic activity of the chromatographic fraction B. And in assessing this, we assess the involvement of endogenous nitric oxide, the involvement of non-protein sulfhydryl compound, and involvement of prostaglandin. For each of these, blocker of these compounds are used. For example, for the assessment of endogenous nitric oxide, N-nitrile arginine methyl ester, which is a blocker of sulfhydryl nitric oxide, was used as a blocker, which is named. And for the that of sulfhydryl, N-ethyl melamine, which is a blocker of endogenous sulfhydryl, were used in order to block this and then assess. So for each of these assessments, 4 different groups were used. The first group, that is 24 animals each. The first group contains the control group, in which there was no, it was only given distilled water. The second group was the one that contained only the blocker. So for example, for that of nitric oxide, NAME, which is N-nitrile arginine methyl ester, was used as a blocker, 70 milligram per kilogram body weight of this. Then for the third group, it was only given, administered only the fraction B. Then the last group was given both the chromatographic fraction and the blocker. After all this, ulcer were then induced using 1 ml of 95% ethanol. And also that was done for assessment of endogenous sulfhydryl using N-ethyl melamine, which is NEM. We have the one that has only the blocker, the control, the one that has the fraction, and the one that has both the blocker and the fraction before ulcer induction. Then the last one was the assessment of involvement of prostaglandin. This was evaluated using a blocker of prostaglandin synthesis. Indomethacin is generally known as a drug that blocks an enzyme that is involved in prostaglandin synthesis, that is cyclooxygenase. So 24 rats was also used. So here we have the control. We have indomethacin that was given to the group as a blocker. Then we have the group that had only the fraction, and we have the group that had only the blocker and the fraction before ulcer was now induced using ethanol. Results and discussion. Table 1 here shows the ulcer index and inhibition in ethanol-induced ulcerated rat treated with the chromatography fractions. The column chromatography of N-butanol extract of Acacia nilotica pod gave 3 fractions, which is fraction A and fraction B and fraction C. And this was what was subjected to in vivo study. So at the end of the day, we have fraction A having an ulcer index of 1.833, which is significantly different from the ulcerated control, which is having about 21.17. However, in comparison with all the fractions, which is fraction A, fraction B, and fraction C, fraction B had a significant reduction when compared with other, which is 0.833, lower than all the reduction that was observed from all the other fractions. And this is translated to be about 96.08 ulcer inhibition, which is the highest among the fractions. Thus, fraction B shows the most promising gastroprotective effects. Table 2 here shows the ulcer index of animals with ethanol-induced ulceration that was treated with a blocker of nitric oxide synthesis. So here we have the ulcer index of the blocker having about 32.83 millimeter, which is significantly higher than the ulcerated control. That means at the end of the day, when the blocker was used, it potentiates the formation of ulcer on the gastric mucosa. However, by the time fraction B is used, we can see that the ulcer index was significantly reduced when compared with that of ulcerated rat or that of all other groups as compared. However, if you look at the combined administration of L-name and fraction B, it does not really change the ulcer index when it is compared with only the blocker, showing that the mechanism of action of this may not be mediated by nitric oxide blocker. Table 3 here shows the ulcer index of animals with ethanol-induced ulceration that was predicted with L-ethyl melamine, which is a blocker of endogenous sulfhydryl group. Here, you can see that by the time the blocker, which is N-name, was used in group 2, which is N-name, we had an ulcer index that was significantly higher than that of the ulcerated group that does not have the ulcerated. However, by the time this blocker was used together with fraction B, which is our fraction of interest, we can see that the ulcer index significantly reduced, meaning that the if the ulceration is being potentiated or the gastroprotective effect is being mediated by the action of sulfhydryl group, we shouldn't have this reduction. Thus, this may not be the mechanism of action that's for the gastroprotection of fraction B, chromatography fraction B. Table 4 here shows the ulcer index and prostaglandin concentration of animals with ethanol-induced ulceration that were pretreated with indomethacin. So we have the ulcerated control. So here we can see that this was significantly reduced, and thus this shows that the mechanism of action could be as a result of synthesis of prostaglandin. In conclusion, fraction B, which is obtained from the column chromatography of N-butanol fraction of the partitioned aqueous extract of Acacia and nilotica bond, had the most effective anti-ulcerogenic activity. And this anti-ulcerogenic activity is possibly by increasing nitric oxide synthesis and endogenous tau group. These are my references.